Today, we're jumping into some really fascinating research that might just hold a new clue for detecting cancer, and it all comes from a paper in the Journal of Materials Chemistry B.
So, let me just start with this question. What if a simple, non-invasive spit test could give us an early warning for one of the toughest diseases out there? I know, it sounds a bit like science fiction, but as we're about to see, researchers are getting way closer than you might think.
You see, the whole idea hinges on this fact. Our bodies are constantly leaving behind little clues about our health, and in the world of medicine, these clues have a name. They're called biomarkers. And when we're talking about cancer, these biomarkers act like big, bright red flags. The runaway cell growth that's a hallmark of cancer often causes really high levels of certain chemicals to show up in our bodies. And yeah, that includes in our saliva.
Okay, so finding the right biomarker is the whole game. The research we're looking at today zeroes in on one in particular. It's called spermine. Now, spermine is a compound that's absolutely essential for normal cell growth. And since cancer is basically a disease of out-of-control cell growth, it makes a ton of sense that watching spermine levels could give us a critical clue. In fact, studies have already shown a strong link between unusually high levels of spermine in saliva and breast cancer, along with other solid tumors.
So finding the clue is one thing, but how on earth do you reliably fish it out of something as messy and complex as human saliva? Well, that brings us to the really cool part of the story, the smart sensor solution. We're talking about building a molecular detective.
And listen, this is not easy. As the researchers themselves put it in their paper, creating artificial materials that can recognize a specific molecule with the same precision as a natural enzyme, that is a huge challenge. Nature is brilliant at this stuff, but trying to copy it in a lab is incredibly difficult.
But they did it. So let's meet the hero of our story, the FEM-MIPI nanozyme. It's this tiny synthetic sensor that has been engineered for one very, very specific job: to find spermine molecules and catch them. So how did they make this molecular detective so good at its job? Well, the secret lies in how they built it, by creating a perfect molecular mold.
The core technology here is called molecular imprinting. Honestly, the easiest way to wrap your head around this is to think of it like creating a custom lock for a very, very specific key. In our case, the key is the spermine molecule, and the sensor, well, that's the lock.
Okay, I know this schematic from the paper looks pretty complex, but don't worry about the details. It's really just showing the step-by-step recipe they followed to build this thing. Let's break down the main idea, because it's actually pretty brilliant.
Right, here's how it works. First, you take your target, the spermine, and you mix it with the basic building blocks of a polymer. Second, you let that polymer form a solid cast all around those spermine molecules. Third, and this is the magic step, you just wash the spermine away. And what are you left with? You have this polymer full of perfectly shaped little pockets, these molecular molds that are an exact fit for spermine and pretty much nothing else.
And just to prove that this whole imprinting thing worked, they also created a control version without using spermine as a template. The difference here is everything. The imprinted sensor, our FEM-MIPI, has what you could call a molecular memory for spermine. The non-imprinted one, on the other hand, is just a generic surface with no memory at all. This is the secret to its amazing specificity.
Okay, so the theory is fantastic, right? But this is science. They had to put the sensor through its paces. The big question is, does it actually work?
First up, how sensitive is it? Well, its limit of detection was a staggering 220 picomolar. Now, that number probably doesn't mean a whole lot on its own, so let me put it this way. That is like trying to find one single, specific drop of water in 20 Olympic-sized swimming pools. It is just astoundingly sensitive. But being sensitive is kind of useless if you can't be selective, right? Can it pick spermidine out of a crowd? The answer is a huge yes. They tested it against 6 other things you'd commonly find in saliva, like glucose and urea. And just look at this chart. You can see the sensor's response just skyrockets for its target, spermidine, that's labeled SPE, while it barely even notices the other stuff. It's a molecular sharpshooter.
This is all great in a clean, controlled lab environment, but the real acid test, that's human saliva, a complex, messy cocktail of all sorts of molecules. So the team moved on to the real-world challenge.
And you know what? The results were seriously impressive. They took saliva samples, added a known amount of extra spermine, and checked if the sensor could find it. The average recovery rate was 86.4%. This is huge because it proves the sensor can work effectively even in a messy, real-world biological fluid.
Let's look at one specific example.
So first, they measured how much spermine was already in the saliva sample. Then, they added 600 nanomolar of extra spermine. And boom, the sensor measured it again and found 533 nanomolar of that added amount. This just shows you exactly how accurate it can be in a practical situation.
So, what does all this mean for the future? Well, this is all about taking this incredible science from a high-tech lab bench and putting it right into our pockets.
The researcher's vision, which you can see in this diagram, is a complete, super user-friendly system. We're talking a disposable sensor strip, a small wireless reader, and an app on your smartphone that gives you quick results. It's a simple, elegant, and really powerful concept.
When you break it down, this technology really ticks all the boxes for a potential game changer. It's designed to be cost-effective and disposable. It's a non-invasive saliva test, no needles. It gives you a result in just 10 minutes, works wirelessly with your phone, and as we've seen, it is incredibly sensitive and selective.
Which leaves us with one final, powerful question to think about. We've all seen how technology has made daily glucose monitoring simple and accessible for millions of people. So, could this be the breakthrough that finally does the exact same thing for cancer screening? The potential is just enormous.